Welcome to Lab in a Box, a program brought to you by Genes in Space to help bring current molecular biology techniques to your classroom. Today we're going to go over the basics of what's in your kit, how to use each piece of equipment, and how to conduct a successful in-class experiment. We'll break this down into 8 main topics: receiving your kit, introduction to pipetting, PCR, restriction digests, preparing for and running gel electrophoresis, interpreting your results, and returning your kit. You can find time codes for each of these sections in the description box below. Let's get started with receiving your kit and going over each piece of equipment. You will receive your loaner kit in a shipment of 2 boxes. The first will contain the food safety lab kits, which will ship on ice. The contents of these kits must remain frozen until you begin the lab. Make sure you put them into a freezer immediately upon receipt. This kit will contain Easy PCR Master Mix, Food Safety Primer Mix, 4 template DNA samples, a restriction enzyme, and a DNA ladder. Store all of the reagents in the freezer until right before use. Take special care with the restriction enzyme, as even short periods at room temperature can affect its activity. The second box will contain the equipment needed to perform the PCR and electrophoresis activities. The case will come packaged in a cardboard box. Make sure you keep this for shipping the kit back when your loan period is complete. You will receive a combination to unlock the case by email, and you should have your equipment checklist ready upon opening so that you can assure you have received all of the necessary components. You will receive 8 H20 pipettes, 1 H200 pipette for instructor use only, 1 H10 pipette for instructor use only, clear and yellow pipette tips, PCR and microtubes, agarose, gel green, and TBE buffer, Clearview spray and a microfiber cloth, 4 gel casting platforms, along with 8 casting trays and combs, 4 blue gel boxes and power supplies, each with a blue base, buffer chamber, and orange cover, 4 mini-PCR machines, each with a USB cord, power supply, and AC cord, an agarose scale, and 4 fold-of-views. You will additionally need to acquire and supply a few things on your own, like distilled water and flasks and beakers. You can find the full list of instructor-provided materials in the instructor manual. Once you have acquired all necessary materials, it's time to familiarize yourself with the setup of the Lab in a Box activity. In this activity, students will examine 2 mock food samples by amplifying a short region of the DNA using PCR and digesting it using a restriction enzyme. They will then examine the resulting DNA fragments using gel electrophoresis. The example scenario is described in more detail in your lab activity packet. The full activity will likely take about 150 minutes or 3 45-minute class periods. We recommend the following structure. Class 1, Genes in Space and Pipetting. Class 2, PCR. Class 3, Restriction Enzyme Digestion and Gel Electrophoresis. If class time allows, you might want to split Class 3 into 2 parts. Micropipettes allow us to accurately measure small volumes. It's important that you and your students become comfortable with using pipettes, as they will be used throughout the lab. There are 3 types of pipettes provided with the kit: 1 H200, 8 H20s, and 1 H10. The students will only be using the H20s, while the other 2 will be used by the instructor to dispense certain reagents. When using a pipette, you should first make sure that you select the appropriate pipette for your volume. For example, the H20 pipette can measure volumes between 2 to 20 microliters. Trying to use a pipette at a volume outside of its range will result in inaccurate measurements and may damage the pipette. Set the volume of your pipette by carefully turning the top dial. Remember that a microliter is just a very tiny drop of liquid, only 1/1,000,000th of a liter. Let's look at the H20. There are 3 digits displayed here. The top digit is the tens place, the second digit is the ones place, and the third digit is the decimal place. So if you wanted to pipette 15 microliters, you would turn the dial so that the numbers looked like this, with a 1 in the tens place, a 5 in the ones place, and a 0 in the decimal place. On the H10 and H20 pipettes, the decimal place is in red or blue, while the H200 pipette does not have a decimal place. Once you have selected a pipette, you will need to add a pipette tip. The tips provided here are loose, so you will want to carefully pick it up by the sides and firmly slip it onto your pipette, just like this. Make sure it is well attached and make sure to always use a tip. Liquid should never enter the main body of the pipette. You will also want to change tips between samples or reagents to prevent contamination. To dispose of a tip, press down on the ejection button over a waste bucket. There are 2 stops on the pipette plunger. When you press down, you will feel a moment of resistance before you push the plunger all the way down. This is called the first stop. Pressing the plunger all the way to the bottom is called the second stop. You should practice stopping at each of these points a few times before pipetting. To draw liquid into the pipette, push down on the plunger until you feel the first stop of resistance. Then, place the pipette tip into the liquid and release the plunger slowly. You can then remove the pipette from the liquid. To dispense into a new tube, place the tip into the bottom of that tube and push down the plunger past the first stop until you feel the second stop. If the tube already has liquid in it, make sure you dispense the volume in the pipette into that liquid to ensure proper mixing. Now pull your pipette tip away before releasing the plunger to ensure that the liquid is not drawn back up into the pipette. It can be helpful to hold the tubes up to eye level so that you can see exactly where you are dispensing the liquid. Take care that the pipette tip is touching either the surface of the tube or another liquid before pipetting, or small droplets may stick to the pipette tip. If you need to mix a solution by pipetting, first push the plunger down to the first stop and place the tip into the liquid. Carefully release and push the plunger, only to the first stop, with the tip remaining in the liquid to mix. Make sure not to create bubbles in your solution. If you get bubbles in your solution, or the solution ends up sprayed on the side of the tubes, the liquid can be returned to the bottom of the tube by gently shaking it. Once you become comfortable using the pipette, you can help your students to become comfortable as well by leading them through one of the optional pipetting exercises in the instructor manual. In Section 3, you and your class will begin the Food Safety Lab activity. Students will use polymerase chain reaction, or PCR, to amplify a specific piece of E. coli DNA in their controls and food samples, so that they have billions of copies of this sequence. In later stages, we will analyze this DNA and determine which food contains pathogenic E. coli. You can learn more about PCR by watching the video in the card above. Before class, use the H200 pipette to dispense 75 microliters of the PCR master mix into a microtube for each group. You should also dispense 50 microliters of the primer mix into a separate microtube. Use an H20 pipette to aliquot 5 microliters of each DNA sample, A, B, P, and NP, into a PCR tube for each group. Make sure each tube is clearly labeled before pipetting. Each group should additionally have at least one H20 pipette and tips, as well as the miniPCR and associated cables. You will also need to supply safety equipment, a waste container, a marker, and a computer or tablet with miniPCR software installed and running for each group. We encourage you to have your students use their own computers if they have them. To save time, you will want to ensure they install the miniPCR software before class. During class, each group should label the tubes containing their DNA sample with their group name before adding the additional PCR reaction components. To each PCR tube, they should add 10 microliters of the primer mix and 15 microliters of the PCR master mix before gently mixing by pipetting up and down 3 to 4 times. Remember to change pipette tips each and every time you pipette to prevent contamination of both the reagents and the samples. Next, the group should open the miniPCR lid by gently pressing on the side tabs and place their mixed reactions inside, pressing firmly to ensure that the caps are closed. Make sure to loosen the screw before you close the lid of the miniPCR. The lid should be closed and tightened only gently with the black knob. Make sure you remember to plug in the miniPCR. Open the miniPCR app on the connected computer. The software may look slightly different on different operating systems, but will have the same functionality. Click on the plus button at the top of the screen, then select PCR. If you wish, you can program and save the protocol before class. Name the protocol, then set the parameters as outlined in the lab activity guide. Make sure that the miniPCR machine is connected to the laptop via USB. Then click Save and Run to load the protocol. The lights on the miniPCR will flash to indicate that the program is loaded. You should now turn the miniPCR power switch to on. Students can click on the Now Running tab to watch a depiction of what is happening in the tube at each step, and to monitor the estimated number of DNA copies present in the tube. To learn more about what is happening in the PCR tube and the importance of each added reagent, you can click on the card above to watch the Genes in Space PCR video. The reactions take about 45 minutes and may run past the end of your class. Once the reaction is complete, carefully release the top of the miniPCR and remove the tubes without touching the metal plate, as it may still be hot. Tubes may now be stored until the next class. If storing for more than a week, refrigerate or freeze the tubes. Next, we'll move on to the restriction digest portion of the lab. We will use a restriction enzyme digestion to distinguish the DNA sequence of the pathogenic E. coli from the DNA sequence of the common non-pathogenic E. coli. Groups should have an H20 pipette and tips, a mini-PCR, 4 empty new PCR tubes, and their PCR reactions from the prior section. In addition, the instructor will need to supply a computer to program the mini-PCR, safety equipment, and a marker. Students should label a new set of tubes for their digestion reactions, 1 tube for each sample, AX, BX, PX, and NPX, and then transfer 15 microliters of each original reaction to the new tubes. For example, they should move 15 microliters of tube A to tube AX. Students may wish to tap or shake down the original reactions before pipetting to ensure all of the reaction is in the bottom of the tube. Do not throw out the original reactions after pipetting. We recommend that the instructor dispenses the restriction enzyme, XMN1, into each new tube. Please keep XMN1 on ice or in the freezer right up until it's time to dispense to the students. Liquid may stick to the sides of the tube. Spin the tube in a centrifuge or flick the tube to collect all liquid at the bottom. Using the H10 pipette and the clear pipette tips, transfer 1 microliter of restriction enzyme into each tube, pipetting up and down gently into the solution to mix, and changing pipette tips in between. Groups may now close the caps of their tubes and place them into the mini-PCR as before. When they open the software, they should this time create a new protocol with a single 37C 15-minute incubation step. You can do this by again selecting New Protocol, Heat Block, and then changing the settings to display 37C for 15 minutes. If immediately continuing on to gel electrophoresis, remove the tubes. Otherwise, perform a 65C incubation step for 20 minutes to inactivate the restriction enzyme, and then remove the tubes and store in a freezer. In this section, we will prepare agarose gels for gel electrophoresis. These gels will allow us to visualize our DNA fragments a little later on. To learn more about how electrophoresis works, you can click on the links in the description below, or click on the card above to watch a video. Prior to gel electrophoresis, we will need to make agarose gels. You may create the gels before class and store them, tightly wrapped and protected from light, in a fridge for up to 3 days. You may also choose to have students pour the gels themselves in class. To prepare 4 gels at a time, dilute 10 milliliters of the supplied 20x TBE with 190 milliliters of distilled water to create TBE buffer at 1x concentration. Place a clear gel casting tray into each larger casting platform and place a comb at the top of each gel. If 2 groups will share 1 gel, you will need to use 2 rows of combs. Make sure you place the side of the comb with 9 teeth down into the casting chamber. To prepare 4 gels, weigh 1.6 grams of agarose using the supplied scale and mix with 80 milliliters of the 1x TBE in a flask or beaker. Gently swirl to mix. Heat the mixture on a hot plate or in a microwave for 40 to 60 seconds until the agarose is dissolved and the solution becomes clear. The solution will be very hot. Use proper protective gloves or mitts when handling. Allow the agarose mixture to cool slightly for 2 to 3 minutes. After this period of time, you should be able to comfortably hold the beaker in your hand for a few seconds to tell if it is cool enough to add the gel green dye. If it is, add 8 microliters of the gel green solution. swirl to mix, then pour 20 milliliters of the mixture into each prepared casting tray. It should reach about halfway up the combs. Allow the gel to solidify for 10 to 20 minutes until firm to the touch, then remove the comb. Gels may now be used immediately or stored in the fridge for up to 3 days. If storing the gels, they should be wrapped in plastic to prevent them from drying out and shielded from light to protect the light-sensitive gel green. We recommend storing them in foil or in a paper bag. In this section, we will use gel electrophoresis to separate our DNA fragments by size and visualize the results of our experiment. Groups should now be prepared to load their samples, both digested and non-digested, into the gels. Each group should be equipped with a blue gel and associated cables, an agarose gel, an H20 pipette and tips, a waste container, their samples, the DNA ladder supplied in the kit, the leftover 1x TBE from before, and a camera phone for documentation. 2 groups may share one gel and blue gel if needed. Place an agarose gel into the blue gel chamber. Fill the clear buffer chamber with 1x TBE until it covers the top of the gel, about 25 milliliters. Use the H20 pipette to carefully add each sample into the wells of the gel. When loading each sample, make sure to dispense slowly into the well. Take care to place just the end of the pipette tip into the well. Do not puncture the bottom of the well, as the sample may leak out. Load the samples in the following order. Before placing the orange cover onto the blue gel box, spray 1 pump of Clearview onto the inside of the cover and carefully spread into an even layer with the blue microfiber cloth. Now place the orange top on the blue gel. Press the Run button on the blue gel box and allow to run for 15 to 20 minutes. The green LED indicator light should remain on for the duration of the run. Darken the room to view the migration of DNA in the gel. Then turn on the blue light. The blue gel should be run for at least 15 to 20 minutes to allow separation of the DNA bands. You may decide to use a camera phone and the fold view to take a picture of the gel after it has run. To find out more about what is happening inside the electrophoresis gel and how it separates DNA by size, you can click on the links in the description below or on the card above to watch the Genes in Space gel electrophoresis video. Your final gel image should look like this. The first 4 PCR lanes contain successfully amplified DNA fragments. The next 4 lanes contain the products of our restriction digest, which will help us to distinguish between pathogenic and non-pathogenic DNA sequences. The piece of DNA we amplified from our samples is 400 base pairs long. When cut by the restriction enzyme, it should be cut into 2 pieces, one that is 250 base pairs long and one that is 150 base pairs long. You can see that in our negative control, the harmless E. coli DNA fragment is 400 bases long, but in the positive control, the pathogenic E. coli DNA has been cut into 2 smaller pieces. As you can see, our space sushi, sample A, has a 400 base pair band and matches the harmless E. coli strain. But the space burger sample, sample B, has 2 shorter bands and matches the pathogenic E. coli sample. This means the space burgers are the food poisoning culprit. If your gel does not look like the sample image displayed here, you can troubleshoot what went wrong by going over the most likely potential pitfalls. If you see only 400 base pair bands in your gel and no 250 or 150 base pair bands, it is likely that your restriction digest failed. This may be due to the restriction enzyme being left at room temperature for too long or not being added to the reaction. If the digestion is inefficient and only some of the PCR amplicons are cut, you may see 3 bands in the pathogenic lanes of 400, 250, and 150 base pairs in length, corresponding to uncut and cut pieces of DNA. It is expected that you will see fuzzy bands at the very bottom of your gel lanes. These are primer dimers, a result of the short PCR primers binding to each other rather than the target DNA. If you see no bands at all in your sample lanes, it is possible that your PCR amplification failed. Make sure that all components of the PCR reaction are added and that the correct number of cycles is performed on the miniPCR device. Seeing no bands in the gel, or in 1 or 2 lanes of the gel, could also be an indication that gel loading failed. If samples are not added directly into the wells, or the pipette tip pierces through the bottom of the gel, they may float away and produce no bands on the gel. If you see no bands and no ladder, it is possible that no gel green was added to the gel, or that the gels were exposed to light during storage. After you have completed your activity, it is time to clean and return the equipment. Clean the orange cover, buffer chamber, casting tray, casting platform, and combs by rinsing them in water and allowing to air dry. Do not expose any additional equipment to water. Once dry, pack all of the equipment into the original case, using your equipment list as a guide. Make sure that the pipettes are returned to their original bag and that the lids of the mini-PCRs are gently tightened by turning the black knob. Leftover consumables such as tubes or pipette tips are yours to keep. Use packing material to fill any empty spaces in the case. Place your completed checklist and the return report into the case and lock it. If you are choosing to share photos of your students with Genes in Space, make sure to also include the media release forms. Place the case into its original cardboard box and affix the return label, emailed to you a few days before your shipment. Finally, drop the box off at a UPS store. Please also be sure to complete the online program evaluation. And that's it. If you have any questions, please do not hesitate to contact the Genes in Space team by emailing genesinspace@minipcr.com or by calling 781-990-8727.